Now I'd like to focus on a signal transduction pathway that involves insulin. Now before we take a look at the details of the pathway, let's discuss what insulin is, what it does, and when our body actually uses it. So let's suppose we just had a meal, and the meal is rich in carbohydrates. And what that means is our body will begin to break down the carbohydrates, the polysaccharides, into their individual units, glucose molecules. And so following a meal that is rich in carbohydrates, we see that inside our blood, the glucose levels will begin to increase. And the rise in concentration of glucose in the blood can actually be very dangerous, very toxic to our body. And so what the body does is it responds by stimulating the beta cells of the islets of Langerhans that are part of the pancreas to release insulin molecules into the blood. And insulin is a small peptide hormone. Now what insulin does is it initiates this signal transduction pathway. And the signal transduction pathway is actually very complicated, very complex, and very extensive. And so what I'd like to focus on in this lecture is actually a small section of this insulin signal transduction pathway that stimulates the absorption of the glucose by the cells and a subsequent transformation of the glucose into the glycogen form. So this is what I'd like to focus on in this lecture. So let's see exactly how that takes place. Now let's begin by focusing on the structure of the receptor that actually binds insulin. So insulin binds onto a receptor we call the insulin receptor. And the insulin receptor actually is a dimer that consists of two identical chains. One chain and a second identical chain. And each one of these chains themselves consist of two individual units. We have the alpha unit shown here in purple and this beta unit shown here in pink. Now the alpha unit is attached onto the beta unit by a disulfide bridge shown here. So we have one bridge shown here and another bridge shown on that adjacent identical chain. Now, what exactly is the function of the alpha and the beta units? Well, the two alpha units, which are, by the way, found on the outside of the cell, the extracellular environment, basically create the pocket, the region of space that binds the insulin. And so that exists on the outside of the cell. Now, these beta subunits not only span the entire membrane shown here, but they also have components found on the inside, the cytoplasmic side of the cell. And these two regions of the beta subunits basically contain a very important section that gives this receptor its activity. And this section contains the tyrosine protein kinase domains. Now, a tyrosine protein kinase is basically an enzyme, a protein that phosphorylates tyrosine amino acids on target proteins and enzymes, and we'll see what those are used for in just a moment. So this is what the insulin receptor actually looks like. Now notice there's an important difference between this insulin receptor and the receptors that we spoke of previously. So for instance, in our discussion on the epinephrine signaling pathway, we said that that pathway actually uses G proteins, and those receptors are known as G-coupled protein receptors. But in this particular case, for the case of insulin, we don't actually use any G proteins. Instead, we actually have protein kinases found within the structure of this receptor. Now, what happens when the insulin actually binds? So insulin is the primary messenger of this particular pathway. And the insulin, shown in orange, binds into this cavity that is created by these two alpha units. And once the insulin actually moves in, these two structures, the alpha units, actually close in. And as they close in, they seal off this region so that the insulin cannot actually detach. On top of that, they also cause these two beta subunits to basically move closer together. Now, as these two beta subunits move closer together, the activation region of one of the beta subunits actually moves into the active side of the the other beta subunit. And this causes a cross phosphorylation process in which both of these beta units are actually phosphorylated in the presence of ATP. And what that does is it activates this insulin receptor. And because of the fact that this receptor itself contains a tyrosine protein kinase, we also call the insulin receptor protein kinase. So, once again, when the insulin primary messenger, a peptide hormone, moves into its cavity that is created by the two alpha subunits, the closure of the two alpha chains causes the two beta chains to move closer together. And this leads to cross-phosphorylation, as shown here and here, that basically changes the conformation of those beta subunits, and that activates those beta subunits. So we see the binding of the insulin on one side of that receptor activates it on the other side. And so that signal can basically be transduced, passed down from the outside to the inside of that cell. Now, what actually happens when these two regions are phosphorylated? So this beta subunit and the other beta subunit. Well, remember, what the tyrosine protein kinase does is it phosphorylates, it attaches a phosphoryl group onto tyrosine residues. And some of these tyrosine residues that are phosphorylated actually begin to act as attachment points for other proteins, namely an important protein known as IRS, which stands for insulin receptor substrate. So one of the phosphorylated tyrosine residues on the insulin receptor, more specifically this one over here, basically attracts a protein called the insulin receptor substrate, or simply IRS. And once the IRS, and in this particular case we call it IRS1 because we actually have another one called IRS2, which exists in a different pathway that we're not going to look in this lecture. But IRS1 basically attaches onto this phosphorylated residue, and once it is attached, because it moves in close proximity to the active site of that activated receptor kinase, this green structure, the IRS itself, is phosphorylated at 4 tyrosine residues. So upon binding, the IRS itself is phosphorylated by the insulin receptor kinase. Now, what is the point of phosphorylating this region? And in general, what exactly is the function of the IRS1 structure? Well, the IRS protein is not actually a protein that activates something in the pathway. Instead, what it does is it functions as an adaptive protein. So IRS molecules are called adaptive proteins. Why? Well, because instead of activating anything, they actually act as attachment points for other enzymes, other proteins. So instead, the phosphorylated IRS, so instead of activating anything, they phosphorylate. The phosphorylated IRS basically acts as an attachment point for a lipid kinase. And remember, lipid kinases are protein kinase that take a phosphoryl group from ATP and they move it onto some type of fat molecule, some type of lipid molecule. And to be more specific, the lipid kinase that this actually attaches to is known as phosphoinositide 3 kinase. So we have the phosphorylation of these 4 residues on this IRS molecule causes this molecule here, phosphoinositide 3 kinase, to actually attach. So the phosphoinositide 3 kinase contains a regulatory region shown here that attaches onto the phosphorylated residue of the IRS molecule. And once this attachment takes place, that causes the active site of this lipid kinase to move in close proximity with respect to the membrane. So this is the active site of this phosphoinositide kinase. And once that active site moves there, it causes the phosphorylation of a specific type of fat molecule that exists within the membrane known as PIP2. Now, when did we see PIP2 previously? Well, we actually spoke about PIP2 in our discussion on the phosphoinositide cascade. And we said that PIP2 stands for phosphatidylinositol 4,5-diphosphate. So this molecule contains a polar region that points towards the cytoplasmic side and a nonpolar region that consists of two tails that lies within the membrane. Now, what this lipid kinase does is it basically attaches a phosphoryl group. It takes a phosphoryl group from an ATP molecule and it places that phosphoryl group onto the PIP2, and that creates PIP3. So PIP2 stands for phosphatidylinositol 4,5-diphosphate, but PIP3 stands for phosphatidylinositol 3,4,5-triphosphate. And so we see that this phosphoinositide 3 kinase phosphorylates the third carbon on this PIP2 to form the PIP3. And once we form the PIP3 basically moves along the membrane and eventually ends up on a protein known as PIP3, so PIP3 dependent protein kinase. And once it binds onto this protein kinase shown in green is able to activate an important effector molecule, important protein kinase known as protein kinase B, also known as AKT. So we see that phosphoinositide 3 kinase phosphorylates the PIP2 into PIP3 that then travels along the membrane to activate a protein kinase we call PIP3 dependent protein kinase, or simply PDK1. So this is PDK1. And once PDK is activated by the binding of the PIP3 to this kinase, it then goes on and activates this structure here, which is the inactive form of protein kinase B, also known as AKT. So AKT1 is activated, and once this is activated, it can basically diffuse across the cell membrane. So this protein kinase B, AKT, is not actually bound to the cell membrane, and it can move anywhere within the cytoplasm of that particular cell. And what this AKT does is, what the protein kinase B does is two things. It actually activates enzymes which are responsible for transforming glucose into glycogen, and it also stimulates these protein transporters to move into the membrane and cause the reabsorption of glucose into the cell. So we see that the PDK1 then activates the protein kinase B we also call AKT, and this kinase is not membrane bound and it can move anywhere around that cell. And it does two things. It stimulates the movement of glucose membrane transporters to that cell membrane, which increases the uptake of glucose from the blood and into the cytoplasm of that cell. And once the glucose is inside the cell, this same AKT also phosphorylates, so activates specific enzymes which are responsible for actually transforming those glucose molecules into glycogen molecules. So the glycogen can actually be stored in cells such as skeletal muscle cells and fat cells and so forth. So we see that the major difference between the insulin signal transduction pathway and the pathways we discussed up to this point is that this insulin pathway actually involves a protein kinase present in the actual structure of the receptor that is involved in this pathway. So before we only focused on G-coupled protein receptors, but now we see that other receptors exist that don't use the G proteins, and instead they use these protein kinases. And in this particular case, it's a tyrosine protein kinase.